Yeah, so thanks for the opportunity to speak today. Drug product, is a passion of mine. I've been in the industry for over 30 years, and drug product is always the last thing anybody ever talks about, right? I've been to so many CMC meetings where drug product is the last topic on the agenda. Never get to it, right? So I'm happy to share some of my recent challenges and innovations that I've found working with several gene therapy companies. So next slide. Okay, so brief introduction. We're going to look at a process flow diagram, talk about the scope of the talk, some general considerations. I'm going to look at a few select process step considerations, and then hopefully have a few minutes for any questions and answers. So this is probably a diagram you've seen in every IND you've ever written before. This is a basic drug product process flow diagram. It's applicable to gene therapy, it's applicable to monoclonal antibodies, it's applicable to anything that's aseptically fill-finished, right? So obviously, if you're having a formulation step, a dilution step, you have to prepare a formulation buffer ahead of time. You have to thaw drug substance. We're going to talk about that. That should be the simplest step in the process, but often it is not. Then we have to compound and mix our bulk drug product solution, sterile filter it. We're going to talk about sterile filtration. We aseptically fill using sterile vials, stoppers, or seals. I have CZ here. I have, just counting up this morning, 6 gene therapy clients. Have 2 products in the pipeline. So out of 8 products, 6 are in the CZ platform, 2 are in glass. So just information for you, if you're still in glass, you probably want to transition over to the CZ platform. Has many advantages we could talk about after. Then, of course, required 100% visual inspection, and then we're freezing again at minus 60. What I considered out of scope for this talk was controlled temperature shipping in general, because 10 years ago, controlled temperature shipping was a concern. Today, you should feel comfortable shipping at any temperature, anywhere around the world, because you could get there in 2 days, in many cases overnight, and there should be no concern. If there is, you're using the wrong courier or transport company. And then I have some bonus late-stage considerations. Obviously, most gene therapy companies are in early stages. Like I said, I'm fortunate enough that I'm working on several programs that are mid and late stage. And before I started consulting for BDO, I worked at Spark Therapeutics for 6 years. We commercialized the first gene therapy for an inherited disease in 2018. So I'd like to share some of those experiences after, if we have some time. So what's the first challenge? The first challenge, finding a CDMO who wants to take your product, right? I have here a CDMO dream, and I say more likely a nightmare. So we're talking about extremely high valued and limited supply drug substance, right? We're talking about very small batches. I've made batches with 80 mLs of drug substance to make a phase 1, gene therapy product. And they're infrequent, they're batches, right? You're going to on board with the CDMO, and you're going to make a batch. You might not make another batch for 2 or 3 years. So definitely CDMOs there's not a lot in this space. There's probably 6 or 7 that I work with in the US, and I'm not sure if there's many more than that. Again, if you'd like to talk about some CDMO options, I'd be happy to afterwards. And what are the first questions the CDMOs ask you when you give them your RFP? They want to know about your product, right? So I say we have limited, and that's my nice way of saying no development information, right? So what's your freeze thaw stability? I don't know. Thaw it once, make it, freeze it when you're done. Estimated thaw times. I don't know. How long does it take 700 mils to thaw in a 1 liter PT bottle? Does anyone know? Anyone want to guess? How about 1 liter in a 1 liter bag? How long does that take to thaw? How about if you thaw it at 2 to 8? How long does that take? Nobody knows. I mean, that's the answer, right? So I mean, it's very, very frustrating, and there's so many variables, right? Are you thawing in a biosafety cabinet? Are you thaw it on a lab bench? What's the temperature of the room? I've had experiences of something that I estimated take 12 hours took 30 hours to thaw. Something that I estimated to take 12 hours took 2 hours to thaw. So it's frustration, but I'll have a little slide on that coming up. What's your allowable time out of environment, right? How long could I keep this at, not the frozen state or at ambient? I don't know. So I tell the CMOs, minimize it, minimize time out. Well, what does that mean? Fill it in 1 day, right? Many of the CDMOs They do not have a one-day process, right? They have an overnight 2 to 8 hold before filling, and I understand that. Depending on the size of your batch, it makes sense. If you're working in other spaces such as monoclonal antibodies, right, they're going to want 144 hours of environmental time out of environment, which is we can't do. How about hydrogen peroxide sensitivity? No idea, right? Don't expose my project to hydrogen peroxide. Do we need to aerate down to lower than 1 ppm? I don't know. Can you? Yes. Does it cost more? Yes. All right, 1 ppm is fine. We're going to put it on stability and find out. What's the light sensitivity, right? I don't know. Protect it from light. Do you want us to process in the dark? No, that's crazy. Protect it from light when it's in the freezer, when you're done with it. We're going to get data and be able to support this later during development. What filter should we use? I don't know. What does the substance guys use, right? Are they using a PVDF filter or a PES filter? Let's keep it the same. Maybe we could save some cost on filter validation down the road instead of paying $100,000 twice for the same document that the filter manufacturers know ahead of time what the answer is. And then finally, processing and hold times, right? Again, we don't know. So document, make sure your batch record has entry for date and time of every step in the process and the temperature, so that after we could go through batch records and determine what were the actual hold times, and information will help for us down the road when we get to later stages. So yes, I have many Custa friends who are CDMO business development directors, and I always try to influence them. So why don't you guys do gene therapy? And they say, no thanks, we're not interested in. That slide there is the reason why they don't want to do it. So that's the first challenge we've got to overcome. So I talked about drug substance thawing and what a challenge it is. Well, not that it's a challenge, it's a simple process, right? It should be the easiest process step that you do. My family likes to follow up with me when I'm person in plant. That's a big part of my job, is overseeing, batch manufacturing, and they call me all the time. They're, what are you doing, Dad? Watching ice melt. They're like, you get paid to watch ice melt? I'm like, I get paid very well to watch ice melt, and the longer it takes, the more I get paid. They're like, really? I'm like, that's correct. So as I mentioned, we don't know, we have estimates of how long things take to thaw, but, you're doing it ambient, you're doing it in refrigerated. We control freeze things, so my solution is, why don't we control rate thaw things, right? Why don't we come in, Monday morning and set the control rate, freezer to work in reverse, be done the next morning at 7 a.m. Go from minus 80 to, 5 degrees, and when we're ready at 7, we're ready to go, right? Makes sense. I mean, and there are units that do that. So I'm going to try to get that invoked somewhere along one of my process steps. Maybe I see a couple of my clients here. Maybe we could try that with your product. All right, I want to talk a little bit about sterile filtration. Again, very, very basic diagram here, right? That's the other thing. A lot of times these very early phase programs are acquired from universities, right? And what are they doing? They're doing this in beakers, in biosafety cabinets. They're using filters that can't be integrity tested. It's a very basic process. And it can be a very basic process when you think about it, right? We saw the process flow diagram. We're pooling multiple containers. When I started at Spark, we had 50 mil Falcon tubes were the bulk drug substance. We had 3 or 4 of those filled, and that was the bulk drug substance that had to be poured into an Erlenmeyer flask, mixed, filtered, and filled with a peristaltic pump with a foot pedal, and then hand crimped. Also, a lot of these programs have cryo vials, screw top cryo vials. Can you believe that? That's what's in the clinic in some of these very early programs. Again, at Spark, we had access to the agency, and they were going to allow us to commercialize screw top polypropylene cryo vials. I was in charge of the program. I said, no way, we're not doing that. And we implemented the CZ platform, 2 mil CZ platform. So the point is, sterile filtration can be very simple, and it also could be very complex. So I'm sure we're all aware of the recent Annex 1 modifications. So our European friends have imposed PUPSIT upon us. So we went from that very, very simple diagram to this very complex manifold, right? And in theory, I think PUPSIT makes a lot of sense, right? We certainly want to ensure that our filters are integral before we filter our product, right? We understand the concerns of potentially, clogging a pore that was not integral with product and then passing after the fact. But you have to have very skilled and experienced operators to be able to manage a PUPSIT manifold like this, which is a single-use disposable PUPSIT manifold. So this is what the setup looks like, right? So we have the bulk substance on the left. We have this manifold. We have Wi-Fi. We have formulation buffer, aliquots. We have our 2 filters in series. We have vent filters all over the place. We have a waste bag, and then we have our bulk product that we're going to fill. So the PUPSIT process is largely, you start with your Wi-Fi aliquot. You flush your filters with Wi-Fi. It goes into the waste bag, right? Then you attach your filter integrity tester to your pressure sensors. You integrity test each filter in sequence to demonstrate that you're still integral. Then you will flush your product or buffer through the filters. We have what's called a blowdown step, where we're going to actually put sterile air through this manifold to try to dry the filters. And we don't want to dry the filters, but we don't want to lose any product, right? So there is going to be some residual buffer, and that's something we have to live with. And then we sterile filter. Then that bag in the upper left then gets hung next to the isolator or whatever type of filling parameters you have, and you fill your product. So I often get the question, right, Steve? I understand, you don't want to waste product, but why are you not flushing with product? Aren't you going to dilute your product with buffer? Yes, I am going to dilute my product with buffer. Am I concerned about diluting my product with buffer? Well, you can be, but if this is an example of one of the last fills I did earlier this year, and if you're diluting your product with buffer, it's 5 mils, 10 mils, right? Is that really going to make a difference at the end of the day? So in this case, I had 2.6 liters of bulk drug substance. It was at a concentration of 2.5 E13, which resulted in 6.6 E16 vector genomes. I do the math. It tells me that I have to dilute that 2.6 dilute the 6. I have to add 2.6 liters of buffer on top of here. So within the batch record, you have allowable variations, right, for the amount of formulation buffer you could add, and it's plus or minus 5% in this case. So it resulted in 200 mils either way. So my point is, I have this variability built into the process. If I do have a little buffer left over at the end of the day, I'm going to be pretty close to my actual concentration, and that's what the bottom result here. This was the actual result from this batch. The target was 1.0 E613 vector genomes per ml, and we hit it dead on. So whether I carried over a little buffer or not, it didn't make a difference in this process. So the other end of the process, right, is drug product freezing. Obviously, I skipped a few process steps. These are just the ones I wanted to talk about today. We have the option: do you want to freeze straight to minus 80, or do you want to do a controlled rate freeze to minus 80? And I don't have a strong preference either way. Again, my point is pick one, but whatever option is chosen, you need to be consistent. And then this was a first fill at this CDMO. I was the person in the plant, and I was observing the process, and I saw them take the bulk of the batch and put it in the minus 80 put it in the controlled rate freezer, right? So that was the part that was going to the clinic. That was the rest of the batch. The samples that they pulled for QC testing, for stability testing, they put in a different freezer and froze immediately to minus 80. And I said, what are you guys doing? I said, your stability samples, your release samples, are not representative of the batch because you just had a different mechanism to freeze. So straighten that all out for future batches, that whatever we're doing, we're freezing everything the same way. Otherwise, what good is your data, right? You didn't just test the data that is representative of your batch. Then I have a couple, last slide here really just talks about some late stage considerations. First and foremost, I very much encourage you to interact with regulators. There's a number of regulatory designations available. Fast track, obviously breakthrough therapy. One of the big ones is CDRP, which is the Chemistry, Manufacturing, and Controls Development Readiness Program. Gives you access to the agencies early in development. Hey, this is going to be my plan for commercialization. This is going to be my PPQ plan. Are you on board with it? It's really a great opportunity. One of my clients did this earlier in the year, and we got really good feedback from the agency. One of the biggest issues is, the whole, 3 PPQ batches, which has become kind of, expected for most modalities. But when it comes to gene therapy, I can tell you it is not the standard. Spark commercialized Luxturna with a 1 PPQ batch approach. I have other clients who've done 2 PPQ batch approaches, and we just requested a 1 PPQ batch approach for this other client, and the agency was like, let's see how your development data goes, and we will take another look at it, as you get closer. So we'll have an opportunity before we execute it to make sure that they're on board. The second one is to embrace regulatory guidance, right? There's so much regulatory guidance out there. And recent regulatory guidance. Of course, the holy grail is the process validation guidance from 2011, General Principles and Practices. Does anyone know when the first ever gene therapy was approved anywhere in the world for a product? And what year? No guesses? No guesses? Well, I can tell you, it certainly wasn't in 2011 when this guidance document was written. So for us to apply these principles, right, certainly, but you'd be surprised. 2012, in Russia, a product was approved. It was a VEGF for peripheral artery disease. So I was shocked. I didn't know that until I looked it up. I know, like what I said, I was at Spark. 2017 was our approval for Luxturna. But the point being is, this is a good document. I reference it every time I write a validation master plan or a high-level strategy document. I think the phrase I use is, this is consistent with the process validation guidance. I don't say we follow it exactly, but it's consistent with. And again, as I mentioned, there's numerous recent and legacy guidance documents. So again, the FDA website is excellent. You can find lots of stuff. And then the final area, of course, the end of the line, labeling, packaging, and shipping validation, right? So if you don't know what that is on the top there, that's a serialized barcode, which is required for commercial products, right? Again, when I was at Spark, these regulations evolved over 15 years, right? The implementation of serialization took forever to finally be adopted, but it is adopted now. We didn't do it at Spark. The product is serialized now. We put in an exemption, never heard back, so we didn't do it. They didn't tell us yes, they didn't tell us no. But the point is, these activities are within the PPQ envelope, right? So we spent so much time with this recent CDRP interaction, writing a briefing book to try to eliminate the plasmids, the starting cell banks, all the things that we wanted to be outside the PPQ envelope. So we put that in the briefing book. We said, clearly this is obviously the drug substance process, obviously the drug product manufacturing process. And their response was, don't forget about packaging and labeling. That's inside the PPQ envelope. We're like, oh God, I'm glad they didn't ask about the front end, because that was our goal. We wanted to make sure that those starting materials were out. And that's an evolving thing too, because I worked with other cell therapy companies where we completely validated, gene editing starting materials, right? So I think that's still a point of contention. Learn the serialization language, right? Most of the folks that I work with have zero experience with commercial products and serialization, right? So it's not hard. There's a few major companies that do provide serialization services. Obviously, all the packaging and labelers do it, right? But you're gonna have to learn what GS1 stands for, what your FDA labeler code is, right? What's a GTIN? What's a global location number? And again, it's not complicated. It's hard to set up. Once it's set up, everything's automated, and it's not a big lift. But you have to have some knowledge in this area. And then the final thing, I know the previous speaker said the same thing, is do not overlook shipping validation. I literally got a call, I think in February, from a gene therapy client who said, we're doing our final review of our BLA, and someone pointed out that we don't have a shipping validation section. Can you write it for us? I said, yeah, of course I could write it for you. I said, I could write anything, but I'll give you a standard approach to shipping validation, but during the review period, they are certainly going to ask you for shipping validation. So And I did, I don't think we've heard back yet on that BLA as to what the expectation was. But I said, yeah, we have to have that done. So don't forget about that. It's end of the line, but it's very important, and has to be done. I think when we asked the agency for leniency on PPQ runs, for some of the process characterization work that we're doing, we have to supplement that with make sure we do everything properly when we can, right? It doesn't cost a lot of money or time to do these end of the road activities, so don't skimp where you don't have to.
Lovely. Can we thank Stephen, please?
All right. Thank you.